Meiosis is a type of cell division that specifically creates gametes. The chromosome numbers are cut in half during meiosis. The main goal of meiosis is to produce 4 genetically different haploid cells. A somatic diploid human body cell has 46 chromosomes, 23 from the mother and 23 from the father. The DNA undergoes a phase of replication, just like during mitosis, called interphase 1. The nuclear membrane and nucleolus are intact, just like in mitosis. During prophase 1, chromosomes are first visible, similarly to mitosis. Additionally, homologous pairs are formed and create a tetrad. Homologous chromosomes have the same DNA, one from the mother and one from the father. The DNA can consist of different alleles. The homologous pairs, or tetrads, undergo a key step in meiosis called crossing over. The maternal and paternal chromosomes swap areas of their chromosomes to ensure genetic diversity in their offspring. Genetic diversity helps ensure that each offspring created is different. In addition, diversity helps protect the human race from completely dying off from illness, for example. Similarly to mitosis, the centrioles release spindle fibers that begin to grow towards the tetrads. The nucleolus and nuclear membrane break down, and centrioles begin to move to opposite sides of the cell. During metaphase 1, tetrads line up along the center of the cell, and spindle fibers attach to the centromeres. The same steps occur in mitosis, but with chromosomes. During anaphase 1, homologous chromosomes are pulled to opposite sides of the cell by spindle fibers. In mitosis, the same events occur, but with individual chromosomes. During telophase and cytokinesis 1, each set of homologous chromosomes separate into a daughter cell, creating a total of 2 haploid daughter cells. The nuclear membrane and nucleolus reappear, just like they would in mitosis. A second interphase is uncommon with some cells, but if it does occur, it is very quick and it skips the S phase, because copying the DNA would defeat the purpose of meiosis, to reduce the chromosome number. In prophase 2, the nuclear membrane and nucleolus break down, spindle fibers form, and centrioles move to opposite sides of the cell. The chromosomes are usually coiled from meiosis 1. During metaphase 2, the chromosomes line up in the middle of the cell and the spindle fibers attach to the centromeres. Sister chromatids split and are pulled to opposite sides of the cell by the spindle fibers in anaphase 2. During telophase and cytokinesis 2, the chromosomes uncoil, spindle fibers break down, and the nuclear membrane and nucleolus reform. This produces 4 haploid genetically different daughter cells, or gametes. Meiosis in a male is very different from meiosis in a female. In a male, meiosis produces numerous gametes, much more than a female would. A male produces more gametes so that there is a higher chance for the sperm to reach the egg and successfully fertilize it. The male division is also much more even than the females. To explain, in a female, meiosis is a very uneven division that produces 1 gamete and 3 polar bodies. The egg is much larger so that it can successfully carry a life. The 3 polar bodies are broken up and destroyed in the female's body.